Hi, I'm Anupama Jha. I'm a senior CIS PhD student with Yusuf Barash at the University of Pennsylvania. Today, I'll talk about RBP-Pokedex. It is an unsupervised deep learning approach for predicting the effect of an RBP's knockdown on other RBPs in healthy human tissues. For a longer version of this talk, check out the iRNA cozy. RNA binding proteins are major drivers of post-transcriptional regulation in cells. RBPs can bind and control many different RNA targets and form complex regulatory networks. For example, RBPs can bind directly to their RNA target or indirectly regulate them through other RBPs. RBPs can cooperatively bind to a target or compete with one another. They can also regulate a target through mutually managing each other's expression or auto-regulate their own expression. RBPs bind in a tissue-specific manner for both splicing and gene expression. However, systematic experimental characterization of the effect of RBP knockdowns across tissues is infeasible. Therefore, we want a model that learns gene expression of RBPs in different tissues. To do that, we train a variational autoencoder on gene expression of 531 RBPs in 47 human tissues. Input to the model is gene expression of RBPs in a tissue sample that goes through a variational autoencoder, and out comes the reconstruction of the same gene expression vector in the input. For testing our VAE, we look at the test samples from 47 GTEx tissues and compare the VAE to a baseline. This baseline is mean gene expression of RBPs computed from all samples from that tissue. On x-axis is variance explained by the reconstruction. Higher values are better, and on y-axis are the 47 tissues. Our VAEs outperforms the baseline and is able to learn RBP gene expression across all tissues. Then we move on to unseen tissues, where our VAE still beats the baseline, even though it has never seen these tissues before. Next, we ask, can we simulate the knockdown of RBPs through these predictive models? We do that by first training a VAE on 2-encode cell line with transfer learning using the GTEx model. Then we simulate RBP knockdown by reducing edge weights of the target RBP neuron in the first layer of our model and reducing expression of the target RBP in the input. Our hypothesis is that a VAE learns the correlation structure between RBPs for gene expression reconstruction, and thus disrupting that structure should simulate effect of an RBP knockdown. We evaluate our simulated knockdowns against 490 RBP knockdown experiments from ENCODE. We measure precision and recall of the overlap of RBP targets between the experimental and simulated RBP knockdowns. Here we compare our model with a PCA reconstruction. We see that our model offers moderate precision in capturing targets of RBP knockdowns, and it offers high recall, meaning that it finds most targets of RBP knockdowns. Next, we will follow up on one RBP interaction found by the model. Our VAE predicts that in HepG2 cell line, UCHL5 knockdown alters gene expression of HNRNPK, and HNRNPK knockdown alters UCHL5 gene expression. This hypothesis is validated by RNA-seq data of these knockdowns. We then think that these two RBPs are directly regulating each other through binding in the 3' UTR. And in line with that, we found E-clip peaks for HNRNPK binding in the 3' UTR of the UCHL5 gene, and found E-clip peaks for UCHL5 binding in the 3' UTR of the HNRNPK gene. Furthermore, we want to know if these RBPs are involved in cooperative regulation of each other's target. We found significant overlap between their knockdown targets, and log2 fold change of these common genes in these two experiments generally agree. Then we look at clip binding evidence of HNRNPK and UCHL5 upon these two knockdowns, and find significant binding of both these RBPs to each other's knockdown targets. Finally, we want to show cooperative regulation by UCHL5 and HNRNPK. Therefore, we show magnitude of altered expression upon both these knockdowns in direct or indirect targets of these RBPs. Genes bound by both RBPs have higher expression change compared to those bound by one or none. These results suggest that there is potentially some cooperative regulation between these two proteins. Additionally, we think that this relationship explains some of the indirect effects of depletion of either protein individually. In summary, we show that we can reconstruct RBP gene expression using a VA in seen and unseen tissues. We can model effects of an RBP knockdown in silico, and our model finds real RBP targets that we validate with ENCODE. And we individually validate that there is possibly cooperative regulation happening between UCHL5 and HNRNPK. Finally, we provide RBP-Pokedex as a tool to the RNA community. With that, I'd like to thank my PhD advisor, Professor Yusuf Barash, my thesis committee, and members of the BioCyphers lab. Thank you for your time and patience. I hope you enjoyed the talk.